In this video, we will discuss about this exogenous expression of HLA-E on the NK cells can inhibit host NK cells response. So, how does this idea comes to the researchers or the author of the paper? So, they have read a paper by Hanson et al. And that shows a optimized expression of customized peptide, which consists of peptide region of different proteins and with some mutations, that is by mutating tyrosine with either alanine or cysteine, they are able to increase the expression of those this customized peptide on the cell surface. So considering that idea, they thought if they already have this customized uh, protein that's for which they can increase its efficiencies. It is customized proteins that contains peptide from HLA G, B2M, and this HLA-E uh, heavy chain site. And if they can optimize this peptide by the same mutations, that is by mutating tyrosine either with alanine or cysteine, so they can check the high expression of this HLA's efficiencies against this host NK cells response. So they are able to find out the tyrosines in the sequence is at 84 position, so they mutated tyrosine with alanine and also with cysteine. So their first experiment is to co-culture expressed this different variant of their proteins, and they call their proteins SCE that we have discussed in the first video of this paper. The first when we are discussing first figure of this paper. Sorry. So the first experiment is to express those different variant of SCE in the K562 cells and go culture them with the host T cells. Where the host T cells, they get it from their donors, healthy donors, and then check the effect on this K562 cells. So first they check the expressions of this different variant of SCE. So they can see that SCE with 584C mutant gives the highest expressions when they are transduced with same multiplicity of infection. It is same MOI. So from these results, it seems like this SCE by 84C is the most prominent choice. And thereafter they co-culture this K562 cells with the, and before co-culturing, what they did is they transduce the cells and sort these populations and then expand it. Then co-culture this K562 cells with the host NK and host NK cells, and then check first the degranulations of this NK using CD107A markers. So when the wild type K562 cells is used, they are able to see maximum degranulations, and it goes down with the mutations, and the least degranulation occurs with this Y84C mutations. And similar results they have seen for this IFN gamma positive NK cells, and indicating that there is higher expressions of this SCE with Y84C mutations gives less immunogenic response. And thereafter they check the cell lysis, and they can see that in the case of K562 wild type without any expressions of the SCE, the cell lysis are high. Cell lysis is high, around 80%, but it goes down as they use the expression of the SCE, the wild type or with the mutant. And next they wanted to check whether they can optimize their choice of peptide that they used for expression of SCE, and they check it for different peptide, and they are able to see that HLA G has the most potent peptide which prevent the cell lysis the most, and they checked it till effector to target ratio 3. And thereafter, once they optimize the conditions, what it is, they use the allogenic NK cells and they expressed this optimized SCE with Y84C mutations, and along with this anti-CD47 antibodies to prevent this NKGD2A signaling pathway, so that they can get the results of the immunogenic response without any effect of this NKGD2 inhibitions. And what they are able to, so they co-culture these allogenic NK cells transduced with shRNA to suppress this HLA-ABC. At the same time, they express SCE of this mutant, it is Y84C, and with this HLA G peptide, and co-culture them with host NK cells, which they get it from a healthy donor. And the result shows that whenever this HLA-ABC signals goes down, they're able to see high degranulations and high IFN gamma. However, when they also transduce those HLA-ABC reduced cells with this SCE of the variant of the mutations of this Y84C, they are able to see the suppressions in the degranulations of NK cells and also the suppressions in this IFN gammas positive NK cells. And when they check the cell lysis, here they also see the similar results, indicating that where the HLA-ABC goes down, the cell lysis is high. However, when it is when it is further expressed with this SCE with Y84C of SCE of the Y84C variant, they are able to see the suppressing the cell lysis also, indicating that the expressions of HLA-E inhibits the NK cell response, whereas the reduced expression of HLA-ABC will cause increased NK cell response. So this is what they finally conclude from this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.